Good afternoon. It's my privilege today to introduce our Wednesday afternoon lecture series speaker, Laurie Glimcher. Laurie is a true daughter of Harvard, having spent virtually her whole adult life moving from Radcliffe College to Harvard Medical School, the Massachusetts General Hospital, back to the MGH and Harvard Medical School, and then to the Harvard School of Public Health, where she is the Irene Heinz Given Professor of Immunology with a clinical appointment at the Brigham and Women's Hospital. You can also think of Laurie as a literal daughter of Harvard. Her father, Mel Glimcher, was the head of orthopedic surgery at the MGH and a recently retired distinguished professor at Harvard Medical School. There is only one lacuna in Laurie's heritage, but it is a crucial one. A 3-year tenure at NIH in the NIAID Laboratory of Immunology, from which we may be so bold as to say one can trace the breathtaking series of accomplishments that mark Laurie's oeuvre. Laurie's best known for her path-breaking work in immunological science. She has led in several important fields, including the molecular basis of class II antigen presentation, the transcriptional regulation of MHC genes, B-cell plasma cell differentiation, and the molecular basis of the differentiation of naive CD4 T cells into the various TH sets and the consequences of dysregulated differentiation. Among her most striking accomplishments is the identification, cloning, and characterization of the master regulatory transcription factor for Th1 differentiation, TBET, and an analysis of its mode of action and the demonstration of the major consequences of its absence. She has done elegant work on the study of plasma cell differentiation with a beautiful determination of the role of XBP1 in this process and an elegant analysis of the unfolded protein reaction in plasma cells. This work has earned Laurie many accolades, including membership in the National Academy of Sciences and in the Institute of Medicine. She served as president of the American Association of Immunologists. It would have been no surprise if Laurie had chosen a seminar topic based on any one of a series of elegant basic to clinical studies of the immune system, including an analysis of inflammatory bowel disease or even of ER stress in type 2 diabetes. But rather, it is bone today. Laurie has taken the approaches that served her so well in studying the immune system and applied them to the analysis of the dynamics of bone. She was propelled into this field by her discovery of SNRI3, I hope that's the right pronunciation, in osteoblasts, and her determination that mice lacking SNRI3 display a profoundly elevated bone mass phenotype due to augmented anabolic bone formation. Interestingly, SNRI3 also controls bone resorption. Laurie's recently undertaken an RNAi genetic screen coupled with an extensive gene profiling analysis to assemble a picture of osteoblast signaling pathways proximal to or instigated by SNRI3. These analyses have provided novel targets in osteoblasts through which adult bone mass may be controlled and have led to the identification of new proteins that regulate osteoblast and osteoclast commitment and activation. Her discoveries, both in the study of bone metabolism and throughout her research effort, have great potential for translation to the clinic and to provide potential targets for drug development. So today, Laurie's lecture has the provocative title, Bound and Determined, Self-Fate in Health and Disease. So join me in welcoming Laurie Glimcher. I'm also instructed to tell you after the lecture there will be a reception in the library, and please all come.
Thank you very much. It's an honor to be here. It's always an honor to be here to give the director's lecture. And I hope you'll forgive me for venturing into the skeletal system, but I've come to find it rather interesting, and I hope that you will find it at least somewhat interesting, and I'll try to make it to say something, what I think about the importance of the skeletal system in human disease. Okay. Strange. Why is that up there? That's like really weird. That happens to be. Somehow you've got a photo up there instead of my talk, which, you know, it's on this as well. These are family members that I love, but I don't know that they're that interesting to the rest of the audience. No idea how that happened. It's on here. It's not on my Mac. It's just on here, which is really peculiar. This is really a first. You know, I think what I'll do is start talking while they fix this. So we happened onto the field of skeletal biology by mistake, which is how a lot of the science we've done has been initiated. We were actually looking, and this is over a decade ago, for the factor that regulates T helper 1 differentiation, which turned out to be Tibet. But before we found Tibet, we found this other factor called SNRI3. One of my postdocs did an RDA subtraction of Th2 cells from Th1 cells to see what genes were only expressed in Th1 cells. And that search did not turn up Tibet, but it turned up this protein that we called SNRI3. Actually, at the time that we isolated it, it had been isolated by others in a thymocyte library screen, and it was called KRC. It was thought to be involved in signal in Ig gene recombination. So we pulled it up, and we did a number of in vitro experiments in the immune system to see whether it might be, in fact, important in T helper lineage commitment. And we seemed to find a number of functions for it in T cells and in macrophages, but we went ahead and made the knockout mouse. And to our surprise and disappointment, the knockout mouse really had no immune system phenotype. There was a very modest decrease in interleukin-2 production because SNRI3, it turns out, acts as a co-activator of Jun and Phos, so of AP1, and can drive the expression of interleukin-2. But other than that, the mouse was really very, very normal. And I was sitting in my office one day with the graduate student, Mark Wine, and the postdoc, Dallas Jones, who was working on the project. And I said, gee, have you really looked at every single organ in this mouse? I mean, every lymphoid organ, there's got to be a phenotype here. Have you looked at the bone marrow? And Mark said, well, that's the funny thing is that we can't really harvest the bone marrow from this mouse. Seems like the marrow cavity is occluded. I said, well, I suggest you walk down the street to Children's Hospital where my dad, who's a skeletal biologist, has his lab, and take an x-ray of the mouse. And they kind of looked at each other like, oh, okay, let's just humor her, Took an x-ray of the animal and saw incredibly elevated bone mass. And that's really what started us down this particular field. And now let's see if we have any slides. No, we don't seem to. So we'll continue. And this initiated an area in the laboratory that expanded over the next several years as we got funding from Merck Pharmaceuticals to begin to investigate the osteoblast and the osteoclast in more detail. And it turns out that skeletal biology is really a very underpopulated field in contrast to immunology, where there are so many talented investigators and the field is large and very, very vibrant. Skeletal biology is not quite there. And It also is a simpler field than immunology. There are a lot of different cells in the immune system, and keeping track of all of them is getting to be increasingly difficult. But in the skeletal system, there are really only a couple of cells. There's the osteoblast that synthesizes bone matrix, and there's the osteoclast that resorbs bone matrix. There's also the osteocyte, and the osteocyte comprises 95% of all cells in bone. It is the mature, terminally differentiated osteoblast that secretes matrix around itself and gets encapsulated in lamellar bone. There, of course, is cartilage as well, and there's the chondrocyte. But that's about it. And not all that much is known about any of these cells. What are you doing here? I mean, I can keep on talking.
Please.
No problem with that? Okay. Fine. So why do we care about the skeletal system?
How do you get to the mirror? Do you know how to get to the mirror?
Excuse me?
You know how to get to the mirror?
What do you mean it's not being mirrored? Where's the display? Can I do displays here? Arrangement?
Tap on it.
Okay.
You got it. You got it.
All right. All right, now we'll go. Thank you very much. So I can now skip. I will now skip some of the earlier slides to say that SNRI3 is a very large, difficult protein to work with. It's a large protein. It's a member of a small family of proteins, SNRI1 and SNRI2. The founding member is the SNRI protein in Drosophila, which is involved in specification of body parts. And there's not much to say about the sequence of this protein. It's characterized by this ZAS zinc finger pair. There's another pointer. Here we go. And what was thought to be a nuclear localization signal, but otherwise it's just a huge unstructured mass. Skip that. So here's what the mice look like at 14 months. This is a picture of a tibia and a fibula. It's your leg. And in a wild-type animal, that's what it looks like. And in the SNRI3 deficient animal, you can see enormously increased bone mass, but the morphology, the shape of the bone is normal. And if you look at 7 months by histologic analysis, you can see that the marrow cavity is entirely ablated in the SNRI3 deficient animal in comparison to the normal marrow cavity of the wild-type animal. But the bone shape is normal. So just a little primer on micro-CT analysis, which is X-ray microtomography, and which gives you a picture, a 3D picture of the structural properties of bone. So what I'll be showing you are two sorts of pictures, one of the trabecular bone, which is located here in the metaphyseal area of the bone. So here is the articular cartilage. This is the growth plate, the epiphyseal growth plate. This is the metaphysis. The shaft is the diaphysis. And this is the cortical bone, which you can see in cross-section here. And when we look at such pictures in the SNRI3 deficient animals, and this is, I think, at 6 weeks of age. So by 6 weeks of age, the marrow cavity is already being filled with trabecular bone, and the cortices have become thicker. And this is that same picture in cross-section. So here's the diaphysis with trabecular bone in the shaft itself. The skeletal system is an important system, as we all know. You need it for mechanical reasons, and you need it for metabolic reasons, since it's the major store of calcium and phosphorus. There's been a lot of conversation recently, primarily from Gerard Carcenti's lab on bone as an endocrine organ. We'll come back to that in a minute. It also serves as a critical niche environment for the development of hematopoietic cells. We've talked about the two players, the osteoclast, bone resorption, and the osteoblast, bone formation. During development, during embryogenesis, you have development of the bony structures. And there are lots of mutations that are present and that have been described that occur, congenital mutations that alter the morphogenesis of the skeletal system. Relatively few mutations that actually affect adult bone mass. Peak bone mass, the function of the osteoclast is balanced with the function of the osteoblast. So these are coupled in vivo, resulting in normal bone remodeling, because every 10 years you completely replace your skeleton. So the continuous remodeling going on, not only during embryogenesis, but all through life. And peak bone mass is in your late 20s. By your 60s, a large, a frighteningly large percentage of individuals have osteoporosis, where there's too little bone, and that indicates and is due to an increase of osteoclast activity that is not accompanied by an appropriate increase in osteoblast activity. So peak bone mass in men and women is in the late 20s, and in both genders declines precipitously over time, particularly in women after menopause. This is a major health problem, bone loss with age. And what we found in the SNRI3 deficient animals was a complete protection from bone loss with age. So by the time, if you're a C57 black 6 mouse, you have this much bone at peak bone mass. And this is where your bones look at 18 months of age as a mouse. And this is what the SNRI3 deficient animal looks like. So not only does it not lose bone, it's actually gaining bone over time. I had not realized until I started reading in this field what an enormous public health issue this is. 55% of people who are older than 50 have osteopenia. That's probably 1 in 2 women and 2 out of 5 men, which makes for a very large number of people with low bone mass. It's actually more common than diseases that we think of as being more exciting or hear more about, such as cardiovascular disease or cancer. It is defined as the structural deterioration and loss of bone tissue. As we know, it affects primarily women, but one is also can induce bone loss through certain medications, steroids being probably the major example. The major cause of morbidity and indeed mortality in osteoporosis are fractures. 1 out of 4 patients who fracture their hips over the age of 50 will die in the year following their fracture. And as you can imagine, given the aging of the population, this has increased in the last 10 years. So that we can look forward in 2020, those of us over the age of 50 have a significant chance of developing osteoporosis of the hip at huge medical costs. Here's the age distribution today. The blue indicates individuals over the age of 65, 20%. This is what it's going to look like in 2050. We've gone from a pyramid in 1950, where there are a heck of a lot more younger people supporting those of us at the top, to what we are today, which is getting to be a rectangle, and going to be a rectangle completely by 2050, where we're going to have a very aged population. So we need to do something about osteoporosis. Osteoporosis is actually not the only disease that the skeletal system is subject to. Another very important indication for new bone formation is in lytic bony cancer metastases and the breast, lung, myeloma, pancreas, many cancers metastasize to bone. These are very painful lesions, and we really don't have very good treatment for them. And finally, there's inflammatory arthritis, which as a rheumatologist I see, and the painful bony erosions and destruction of bone that accompany rheumatoid arthritis and other inflammatory arthritides, again, with very little treatment for them. So there are treatments for osteoporosis, and they almost all target the osteoclast, the bone resorbing cell, so-called catabolics. And the ones that you know about most are the bisphosphonates, like Fosamax. There's also, of course, estrogen replacement And most recently, Amgen has produced denosumab, which is an antibody to rank ligand, a cytokine that is responsible, the primary driver of osteoclastogenesis. There is one anabolic drug that increases bone formation, that's parathyroid hormone, which is an injectable that's been shown to form new bone, at least for a while. The issue there is the potential for osteosarcomas. So there's a desperate need for new anabolics. And we began to ask, why? Why do these mice have such high bone mass? Which cell type does this map to, and what's the etiology? It's not a developmental anomaly. I think I mentioned that earlier, because you don't see this phenotype appear until about 2 weeks of age. And at about 2 weeks of age, you can see now, colored in red, the increase in trabecular bone mass beginning. There are lots of beautiful work that's been done in developmental biology labs on factors that regulate skeletal morphogenesis, but there really aren't a lot of factors known that regulate adult bone formation. And the process of remodeling of your skeleton begins with, let's say, a microfracture. And we all have lots of microfractures all the time. There's a microfracture, and the precursor of the osteoclast, which is a monocyte, comes in and starts resorbing, turns into an osteoclast and starts resorbing the damaged bone. It is followed by the pre-osteoblast, which derives from a mesenchymal stem cell, which comes in and starts to lay down that bone, continues to differentiate, and continue to form new bone, mineralizes, and becomes an osteocyte. So that's bone remodeling. This is normal bone remodeling when osteoclasts are balanced by osteoblasts, and this is the kind of remodeling you see in osteoporosis, where you have too much bone resorption, not enough bone formation. So you can halt bone resorption with the bisphosphonates or with denosumab. The problem is, because this process is usually coupled in vivo, you end up reducing bone formation eventually. So you can stop bone resorption, but there's no way to get osteoblasts to make new bone. And in fact, bisphosphonates cause them to make less bone. Now, what do these mice look like? We have high bone mass in the long bones in the vertebrae in both genders. It occurs postnatally, and there's an age-associated progression. In biochemical and cellular studies, we mapped the phenotype to the osteoblast and showed that there was, in the absence of SNRI3, there's an increased bone formation rate, increased activity of the osteoblast, and, as I'll discuss, an actual inhibition of osteoclastogenesis. So in the SNRI3 deficient animals, we have a lot of bone formation, and it's proceeding at a faster pace than bone resorption. And it's good bone. It's not woven, unstable, biochemically unattractive bone. It's lamellar bone. It's woven. There is no ectopic mineralization. There are no osteosarcomas, and we've had many of these mice for more than 2 years. The joint is normal. There are no metabolic abnormalities. And the mineral density distribution is also normal. Biomechanically, you can do 3-point bending tests and show that these animals have increased bone strength, which shouldn't be a surprise. Importantly, the heterozygotes have a phenotype, which tells us that if we could inhibit SNRI3 by only 50%, we would see a measurable effect on bone mass, and we'll come back to that. This is what it looks like. This is an experiment in which you label mice with calcine, which is a dye that intercalates in mineral, and you label them twice at day 0 and day 7, and then you see how far apart these two lines are, and that gives you your bone formation rate. So here's the wild type, and here's the SNRI3 deficient. Increased bone formation rate, and we look at parameters like bone formation rate, mineral apposition rate, mineral surface over total bone surface, osteoblast surface over total bone surface. And because SNRI3 controls genetic programs in the osteoblast, in its absence, we see an increased transcription and expression of bone matrix proteins like bone sialoprotein, osteocalcin, osterix, and another transcription factor that's also important in the osteoblast. So these are overactive osteoblasts. This is what they look like. This is a wild-type animal where we have a reporter, a GFP reporter driven by the osterix promoter, which is expressed in osteoblasts. So in green, you can see this is a calvaria, the skull of an animal. Blood vessels are in yellow, bone is in blue, and the osteoblasts are in green. So you can see these little dots, and you're looking at the periosteal and the endosteal surface of the bone. So the periosteal surface is the outside, and the endosteal is the inner layer. Now look at the SNRI3 deficient animals, and I think you can notice a couple things. One is that there are a lot more osteoblasts, and the second is that there are osteoblasts on both the periosteal and the endosteal surfaces of the skull. So in the SNRI3 deficient animal, we have increased osteoblasts. So it controls anabolic bone formation. That's where its function maps to, and you can see that by doing histomorphometric analysis as well as micro-CT. But what's the mechanism by which it can? And we have worked on this problem now for 4 years and are finally, I think, going to be able to crack the code. Certainly, we have obtained by over the last several years, some new insights into how bone mass is regulated by SNRI3 proteins. And what I'd like to cover, if we have the time, if not, we'll just do the first two. I want to discuss where SNRI3 function maps in vivo, the function of SNRI3 in the osteoclast, and how we can target SNRI3 in collaboration with Merck to augment bone formation. Now, the reason we were interested in the site of action of SNRI3 was because of a series of recent papers, very high-profile papers from Gerard Carcenti's lab, where he suggested that bone is an endocrine organ and bone is a metabolic organ as well. And SNRI3 expression is widespread. It's expressed everywhere. It's actually very high levels in brain and thymus. So its distribution is ubiquitous. But Carcenti has described several instances in which bone mass is controlled not by osteoblasts or osteoclasts, but is controlled by the brain or is controlled by serotonin produced in the gut. And we felt is very important to figure out where SNRI3 was active. So we bred the conditional, we generated a line of conditional SNRI3 knockout mice and bred it onto some tissue selective Cre lines. We looked at it in an early PRX1 Cre, which is expressed in limb mesenchyme, in an osterix Cre, which is expressed in the osteoblast, collagen 2 Cre, which is expressed in cartilage and the growth plate, and then in a nestin Cre to ask whether its function might map to the central nervous system. And we did that because Gerard had clearly shown that, for example, leptin can inhibit bone formation through the hypothalamus, controlling bone mass outside of the skeletal system, and had described a mechanism that's dependent on serotonin, where endocrine cells in the gut secrete serotonin and hence control bone mass. But we were not able to identify an extrinsic location for SNRI3 activity. When we looked at the nestin Cre, which deletes in the CNS and the peripheral nervous system, we saw no difference in bone mass. So it presumably maps to bone, and we then set out to look at these different Cres and ask what happens. So the PRX Cre deletes in early limb mesenchyme, and it's early limb mesenchyme that mesenchymal cells come from and differentiate into osteoblasts. And the phenotype was very strong. When we delete SNRI3 selectively in limb mesenchyme, we're looking at bone volume over total volume, trabecular thickness, so the thickness of each of these trabeculi, the number of the trabeculi that you can see, and the cortical thickness, which is this. And I think it's quite apparent that you can recapitulate the phenotype by deleting SNRI3 only in limb mesenchyme. We asked whether, at what stage of differentiation you could delete SNRI3 and still see this phenotype. So we deleted it with the Osterix cree, which deletes in the immature osteoblast. And again, the phenotype is recapitulated. It recapitulates the germline knockout phenotype with increased bone volume over total volume and so forth. And again, we can see here this nice SNRI3 gene dosage effect. So here is the wild type animal, it's the cortical. Here is the heterozygous animal missing only one allele of SNRI3 in osteoblasts, has increase in cortical thickness, and you can see in here some trabeculi beginning to form, and here's the full knockout. Now, I'll just say that we've also deleted it in the osteocyte using the DMP1 cree, and again, recapitulate the phenotype. So it's working all along this pathway. But how far back does it go? We know that we can recapitulate the phenotype if we use a PRX cree, but what if we delete it in this cell type that's been called the osteochondral progenitor, which is still very poorly defined? Well, you can delete it in that cell using a collagen 2 cree, and to our surprise, that also recapitulates the germline phenotype with increased bone mass and cortical thickness. So, what we think is happening is that SNRI3 is clearly a repressor of bone formation, and it can act very early to drive the osteochondral progenitor cell into the chondrocyte, into the osteoblast lineage, if we don't have it. It's a repressor, so without it, we drive this chondral progenitor into the osteoblast lineage, and with it, we're going to drive, it's going to repress bone formation. And indeed, if we overexpress SNRI3, we see decreased bone formation. This is interesting because it gives us a handle, since osteochondral progenitor cells are greatly increased in these animals, of figuring out what this osteochondral progenitor does. It's certainly known to contribute to osteoblast formation during embryogenesis, but its function in adult bone formation is unknown. And we are hopeful that this will enable us, and in fact, we have been successful now in identifying some new markers that identify this cell. To summarize, we can recapitulate the germline phenotype with any of these crees, but not with the nestin cree, so the function clearly maps to bone, and it clearly operates somewhere in the progenitor precursor stage, but also in later differentiation. So its role in bone formation is at the pre-osteoblast, the osteocyte, and also the chondral progenitor cell. Therapeutically, though, we need to know whether deletion of SNRI3 in the adult can result in increased bone mass, because we're not going to be treating individuals in utero with SNRI3 inhibitors. And so we made, with the help of Merck, the SNRI3 inducible knockdown using hairpins that we had validated to target SNRI3 very well. And this is a doxycycline inducible knockdown. And in this animal, so we generated these inducible knockdown mice. We fed them with doxycycline for 6 weeks in this experiment and asked whether we saw any difference in bone mass. And here's the result. So this is an animal that's been fed with doxycycline and has partially deleted SNRI3. This is a partial deletion with a siRNA knockdown. And you can see measurably increased bone mass with 6 weeks of treatment. We repeated this in 22-week-old mice, put them on a dox diet for 6 weeks, did micro-CT and histomorphomatic analysis, and again, a very nice increase in BV over TV, bone volume over total volume, and in cortical thickness. And that is shown quantitatively here, where we're looking at the osteoblast surface over the bone surface, osteoclast surface, and SNRI3 expression levels. So we're only getting about a 30% decrease in SNRI3, but this is accompanied by a substantial increase in bone mass that would be meaningful clinically. We're in the process of doing what is considered the gold standard for osteoporosis research, which is ovariectomy, postmenopausal osteoporosis, to see whether we can prevent, if we delete SNRI3 prior to ovariectomy, can we prevent bone loss? And if we do it post-ovariectomy, can we actually restore some bone mass? And that'll be important. Let me move on to the second story, and that is an unexpected function of SNRI3 in controlling the other major cell type, the osteoclast. And I like this slide, which I took from a book from Tim Arnett at UCL, of a trabecular bone element being eroded by osteoclasts. So here's trabecula, and you can see that it's looking pretty ratty here. And this is that same trabecula from an elderly woman. This is a lumbar vertebrae having been completely severed. We learned in medical school that bone formation and bone resorption are always coupled. And it's true that many mice who have increased osteoblast activity also have a compensatory increase in osteoclast activity. And these processes should be coupled in vivo. And that's really the problem with some of the agents we use to treat osteoporosis, is that they are coupled. And so if you decrease osteoclast activity, you're going to decrease osteoblast activity. And the question is, all right, what's the status of osteoclasts in the SNRI3 knockout? They ought to be increased. We ought to have a compensatory increase in osteoclasts. But in fact, we don't. When we look at serum markers of bone resorption, such as PYD, and this is a cartilage, a connective tissue fragment, we actually see a decrease in evidence, in indices of bone resorption. When you look histologically at osteoclasts, and it's not a very good picture, this is the whole calvaria, the skull, stained in red. This is trap staining, which stains osteoclasts. You can see osteoclasts in the wild-type animal, but you really don't see them in the knockout animal. And when you look under light microscopy, here's the diaphysis of a SNRI3 deficient animal, and there are lots and lots of bone and osteoblasts here, but there are no osteoclasts. So why is this? We know that SNRI3, although it is expressed at low levels in osteoclasts, doesn't really do anything to the osteoclast itself, because we can do chimeric experiments in which we transfer wild-type or SNRI3 deficient bone marrow into irradiated mice, and we know that these osteoclasts are intrinsically normal. So it's not intrinsic to the osteoclast. So it must be a consequence of something that SNRI3 does in the osteoblast that affects the osteoclast. And what is that? So we grew normal osteoclasts on SNRI3 knockout osteoblasts, and we showed that they had a defect in osteoclastogenesis, which led us to believe that there must be a factor secreted from the osteoblast that is required for osteoclastogenesis that is impaired in the absence of SNRI3. And to address that question, we did lots of microarrays from wild-type and knockout bones, and some osteoblast clones that we made that either over or under express SNRI3. And we found a whole bunch of genes that were regulated by SNRI3 in vivo and in vitro. But here's the answer. It turns out that SNRI3 actually controls the transcription of the major osteoclastogenic cytokine, rank ligand. It's the culprit. And its levels are reduced in serum, in bone tissue, and in osteoblasts. And we figured out how it acts at the molecular level. Turns out SNRI3 associates with CREB, transcription factor, and acts as a co-activator, which is the same thing it does for JUN and FOS, but very different than what it does in the osteoblast in controlling bone anabolism. Interestingly, it activates it in osteoblasts, but this is a cytokine that's aroused a lot of interest recently. It's in cartilage, it's in T helper 17 cells, and it doesn't affect, SNRI3 deficiency has no effect on rank ligand levels in those other tissues, so it's very specific to the osteoblast. This is a nice result because it's a double whammy. SNRI3 deficiency, inhibition of SNRI3 will increase osteoblast activity, but it'll actually decrease osteoclastogenesis, which is to uncouple bone formation and resorption. Is this really important in vivo, though? And to ask this question, we teamed up with Ted Gross at the University of Washington, who has a botulinum toxin-induced paralysis model. You inject the mice with Botox, and the wrinkles go away, and they're paralyzed, and they can't move their limbs. And if you can't move your limbs, you very rapidly lose bone, primarily through increased osteoclast activity. And this is what it looks like in a normal animal if you inject them with Botox. You rapidly induce bone loss. But the SNRI3 deficient mice are resistant to this approach. So their limbs are paralyzed, but they do not lose any bone. In fact, they actually continue to gain bone over the several weeks that this experiment is performed. So here's the wild-type animal losing bone, and here's the SNRI3 deficient animal. It's actually gaining both trabecular and cortical bone. And the conclusions are summarized here. I won't read them to you, but we think it makes SNRI3 an even more attractive target because we will uncouple bone formation from bone resorption in vivo. So clearly the reason we were working with Merck, we had a research collaboration with Merck, was because they felt that this was a really exciting potential target to develop therapeutics. That if we could find small molecules that inhibit SNRI3 even by 30%, we should have a measurable effect on adult bone mass. But this molecule is large and ugly and shapeless and is pretty hard to target. So we decided we had better dissect the molecule, and one of my postdocs, Jae-Hyuk Shim, has made literally hundreds of mutants in this molecule, and in fact identified an 8 amino acid stretch in this protein that's 280 kilodaltons, whose mutation led to this phenotype. We know that if we overexpress SNRI3 in human mesenchymal stem cells or in primary calvaria osteoblasts, we repress osteogenesis and mineralization. This 8 amino acid stretch, and in particular, the mutation of 3 lysines to alanines within this 8 amino acid stretch abolishes SNRI3 function in vitro, and it does not inhibit bone formation in vitro either. So this narrowed the function of SNRI3 down to 3 amino acids. And so we made a knock-in mouse where this triple lysine to alanine mutant was made, and we recapitulated a good part of the phenotype. So here is the knock-in, increased bone mass. You can see that here as well. You can see the trabeculi in the shaft and increased cortical. This is not as dramatic a phenotype as the germline knockout or the complete knockout. And this indicates to us that although this region is clearly very important and something we want to focus on, there must be other parts of SNRI3 that make some contribution to its function in controlling bone mass. This is the histology of the knock-in. You can see the trabecular bone already forming here in the metaphysis. One mechanism that we know, so step back for a second. We spent several years trying to figure out how SNRI3 controls the osteoblast activity. And as I said, I think we're close to the answer now. One mechanism is clearly by decreasing protein levels of a master regulator of osteoblast differentiation called RUNX2. So RUNX2 is absolutely critical in the differentiation of a mesenchymal stem cell to an osteoprogenitor cell and then to an osteoblast that synthesizes bone matrix proteins like type 1 collagen and osteocalcin. And we know that SNRI3 somehow reduces the activity of RUNX2. It actually reduces protein levels of RUNX2. We don't know how that occurs, but that's clearly one mechanism by which SNRI3 reduces bone mass. But the phenotype is so dramatic that we don't think that can be the only mechanism. And in fact, SNRI3 interacts with many of the proteins that interact with RUNX2. So SNRI3 interacts with many of these proteins and many others as well. And we believe that RUNX2 is certainly not the only mechanism or the only SNRI3 substrate. We're closing in on what we think is another mechanism, but we don't have final confirmation of it. Given that SNRI3 was tough to target, we said to ourselves, well, let's take a look at all the major signaling pathways in the osteoblast that stem from or control SNRI3. Maybe we can get a handle on another pathway that's druggable, that intersects with SNRI3. And this required, and this is really where the collaboration with Merck was very, very helpful, because with them we took many different approaches to identifying and manipulating these different signaling pathways. So we looked for SNRI3 regulated pathways. We identified SNRI3 associated proteins. We looked at the phosphoproteomics of SNRI3 deficiency. And using genomics and proteomics, RNAi approaches, genetic mutants, genetic mouse mutants, and small molecules. We took all of these approaches to identify SNRI3 influenced signaling circuits. The first approach was transcriptomics, where we basically looked at bones from wild type or SNRI3 deficient animals, or we looked at primary osteoblast cultures, or we profiled osteoblast clones where we'd either overexpressed or silenced SNRI3. So we took a number of different kinds of tissues, and we're looking for those genes that have the right pattern. So they are reduced, they're reduced in the presence of ectopic SNRI3 and increased in its absence, or vice versa, where the overexpression is the opposite of the knockdown or the knockout. And doing that, we found a number of different genes, in fact, 356 genes who had the proper antiregulation. So that's a lot of genes to work with. So we said, okay, let's look at those genes that also overlap with the in vivo profiling of whole bone, as well as these primary osteoblast cells. And we took those 150 genes and asked which ones are druggable. Can we identify nearest neighbors of those genes to expand our list that might be interesting? Are they expressed in osteoblasts? Or have any of them been identified in genome-wide association studies as conferring risk for low bone mass? And we went through this exercise and ended up with about 220 genes, and we said, okay, let's do an RNAi screen on them. Let's silence these and see what's the phenotypic outcome. And the screen we use is either primary Calvera osteoblasts or human mesenchymal stem cells that can be differentiated into osteoblasts. But we're really asking, if we knock down any of these 220 genes, do we get spontaneous osteogenesis in human mesenchymal stem cells? And our readout is both alkaline phosphatase activity, which marks bone, there's a bone synthetic protein, or mineralization, which you can interrogate by calcine labeling. So we did that, and you can see the results. These are genes of interest here. This is a calcine labeling experiment, and this is one positive clone. This is the mesenchymal stem cells under non-differentiating conditions. They don't mineralize, they don't make bony nodules, but if you knock down this particular gene, now you get a lot of mineralized nodules. And we picked up some known genes, which was good. So here are the gene categories that scored in the RNAi screen. I'll show you them here. There's still a lot of them. And now is time to add in small molecules or chemical genomics. So we have gene profiling, RNAi screen. We've also put in here the phosphoproteomics and the interaction, the protein interaction experiments we did, and now we do a small molecule screen. And for this, we designed a reporter cell line, which Merck used to screen a subset of one of its libraries to identify small molecules that promote osteoblast differentiation. We used their small molecule library and identified a number of compounds that targeted some of the genes we identified in the molecular profiling. And so we've gone to the next step and said, all right, we've narrowed it down now to a smaller number of genes. Let's start looking at genetic mutant mice that lack these genes. And I'm going to end with this little story on one of those genes, which is DCAM kinase ligand 1. And this is the work done by Vivo, a postdoctoral fellow in the laboratory with Dallas Jones, now an assistant professor. This is a double cortin and CAM kinase-like protein that has been mainly studied in the nervous system by Chris Walsh. It's a microtubule associating protein. It promotes polymerization of microtubules. It has a kinase domain, although the substrates for that are not known. So we got the DCAM kinase ligand knockout mice from Chris Walsh, and lo and behold, they had increased bone mass. So this strategy, this kind of enormous multi-pronged strategy, can lead to the identification of genes that are somehow on this pathway that may be or may not be more druggable. So increased bone volume and increased bone formation rate in the DCAM kinase ligand knockout animals. Vivo established the mechanism by which this works. It turns out that DCAM kinase ligand suppresses RUNX2 transcriptional activity. And so we asked whether that was really true by doing genetic complementation in vivo. Now, RUNX2, which was identified by Gerard Carcenti, is really the master regulator of osteoblast differentiation. In mice that are heterozygous or humans that are haploinsufficient for RUNX2, mutations in one allele of RUNX2 develop a disease called cleidocranial dysplasia, which is characterized by low bone mass, hypoplasia of the clavicles, and delayed closure of the fontanelles. So we generated RUNX2 DCAM kinase ligand double deficient mice because we're asking, can the absence of DCAM kinase ligand in any way rescue this cleidocranial dysplasia phenotype? And it partially rescues this phenotype. Here is a wild type animal. This is an animal that's lacking DCAM kinase ligand but has normal RUNX2, and you can see it's longer and it has slightly, I don't think you can see it from this, has increased bone density. Here's a cleidocranial dysplasia animal with the malformed skull. It's shorter, has low bone mass. And here is what happens to this mouse if you knock out DCAM kinase ligand. So we're getting partial rescue of this phenotype. And you can see that in the skull as well. You don't have complete rescue, but you have partial rescue of the fontanelles. And now the clavicle, which is gone in the RUNX2 heterozygous animal, has been partly restored if you delete DCAM kinase ligand. And the molecular basis for this is the regulation of the cell cycle by DCAM kinase ligand 1, which affects the phosphorylation of RUNX2. And this particular phosphorylated serine inhibits RUNX2 activity in osteoblast differentiation. And let me stop now because it's 4 o'clock. To summarize, we've identified serendipitously a protein, a large protein, that seems to control adult bone mass. The bone looks good. It's morphogenic, genetically normal. And we know that if we can inhibit levels of SNRI3 by even 30%, we get a very measurable effect on adult bone mass. One target of SNRI3 is RUNX2. It is not the only target. We know that SNRI3 function clearly maps to the osteochondral progenitor and to the osteoblast lineage. So although we have not completely identified the mechanism through which this protein is acting, I think we can take some comfort in knowing that combining all of these approaches has allowed us to identify additional pathways that impinge on SNRI3, some of which may be more druggable. And most recently, we have developed a new screen for SNRI3 to allow us to identify compounds, and this is been funded now by the MLPCN program at the NIH, that will enable our lab and the Broad Institute to do a screen of 400,000 compounds using this novel reporter assay we have that's going to read out levels of SNRI3, because when it's all said and done, SNRI3, I think, still remains the best target, and if we could reduce its levels by a small molecule, we would have a measurable effect on bone mass. And so the assay we've designed is designed to pick up such compounds. We have identified 2 compounds in the reporter assay that reduce reporter activity, and those 2 compounds reduce endogenous levels of SNRI3 and increase osteoblast formation and activity. So we're hopeful, neither of those 2 compounds are druggable, but we're hopeful that the bigger screen that we now can do thanks to the NIH funding will allow us to go through the library of 400,000 compounds and find some good small molecules. I particularly want to acknowledge Dallas Jones, who has been in this project from the very beginning and who's responsible for really almost all of the data that you're seeing here. He was a postdoc in the laboratory and has just been promoted to assistant professor at Harvard. Other key members of my bone team, Mark Wine, who was in at the beginning of this project, Tony Alaprantis, now an assistant professor, and 2 postdocs, Voiguo Zhu and Jay Shim. We've had wonderful collaborations with Merck, a truly collaborative research project with them. And we've had help from my dad's laboratory and Håkan Hofstetter in Vienna, and then several other people at the Broad. Thank you.
Just a short question. Maybe I misunderstood, but it seemed to me you mentioned that conditional knockout of SNRI3 in osteocyte did have a phenotype.
Yes.
And it also high bone mass?
Yes.
How can you explain that?
Well, the CRE that we used is the DMP CRE, which has been thought to be expressed, this is work from Linda Bonewall's lab, only in osteocytes. But it appears from more just data we've heard by word of mouth that it may actually be expressed at some level in the osteoblast as well. But I think it speaks to the osteocyte as a very active component of determining bone mass. So osteocytes are probably the cells that respond to mechanical load. And for example, another major pathway in the osteoblast is the beta-catenin Wnt signaling pathway that connects with the frizzled receptor LRP5. And an inhibitor of that pathway is a protein called Sclerostin. Amgen is working on an antibody to Sclerostin, which actually is very effective in increasing bone formation and it maps to the osteoblast. Now, Sclerostin is thought to be expressed only in the osteocyte. So clearly manipulating gene expression in the osteocyte can affect.
Sure, but is there any, that was my next question, is there any effect of SNRI3 deletion? Is there any effect of SNRI3 deletion on Sclerostin expression?
No, there is not. There is not. It's not acting via that pathway. The SNRI3 pathway seems to be distinct from the Wnt beta-catenin Sclerostin pathway. We don't see any alteration in Sclerostin.
Thank you. That was really interesting. I was wondering what happens if you have a pathological fracture? Do they heal normally?
That's a great question in the SNRI3 knockouts. We're actually doing those studies even now. We have a fracture model. that's very robust thanks to Vicki Rosen at the dental school. And we are looking to see whether fractures, it's kind of hard to fracture the SNRI3 knockout bone, whether fractures will heal more rapidly and will heal normally without, we don't see any ectopic bone formation in the knockouts, but it's an excellent question. And the other model we're looking at is a model of inflammatory arthritis to see if we can prevent or heal bone erosions by inhibiting SNRI3.
Laurie, beautiful story. You mentioned in your introduction that this was found originally in a subtraction between Th1 and Th2 cells. So can you
We
tell
found
us?
it there, but it was found before by a group in Ohio doing screening a thymocyte cDNA library for proteins that bound to an to a recombination signal sequence, actually, which turns out to be irrelevant, but.
So my question is, does this also play a role in the immune system? And if so, what can you tell us about its role there?
As far as we can tell, SNRI3 deficiency leads only to a modest decrease, modest and transient decrease in interleukin-2. It does interact with GIN and FOS and can act as a co-activator, so AP1-driven genes like IL-2 or CD69 are very modestly affected in T cells. We thought it would have a function in macrophages from all the in vitro studies we did, but that turned out not to be the case. We cannot find a phenotype in the immune system. Now, that being said, in collaboration with SNRI2, another family member, it can further exacerbate a severe thymic phenotype. So SNRI2 deficient animals have a severe defect in positive selection. It's actually a defect is in the negative selection to positive selection transition. And so you have very few mature T cells in SNRI2 knockout animals. If you make a double knockout, you get 100% ablation. So it can contribute to thymic development.
And now that you know about connections to things like rankle and so forth, can you look for other effects through those molecules?
It affects rankle ligand only in the osteoblast, not in T helper 17 cells and not in cartilage.
But Laurie, so rank ligand in the thymus is necessary, is the signal that induces the formation of medullary epithelium,
Right.
to form into a medulla. And have you ever looked in the thymus by histological section to see if you have an organized medulla in the SNRI3 knockout?
I cannot remember whether Tracy Staten, who worked on SNRI2 in the thymus, looked at the histology of the thymus or not, because she looked at both the single and the doubles mutants. I mean, thymic development is normal, and
But this is
the response to, infection with Leishmania is normal in SNRI3 knockout animals. I'd be surprised if there were any defect, but it's certainly worth a look.
Yeah, so this is not so thymus site development is normal, is what you're saying.
Right.
The question is whether the thymic epithelial cell development is
Right.
normal.
And I think it's certainly worth taking a look. My guess is that it won't control rank ligand in the thymus as it doesn't control it in T cells.
Well, Laurie, thank you very much. Please join us in the library for a